spread their, or inoculated their bacteria over the actual potato. That's on page 63 where they start talking about that. Aseptic technique, it also goes through and explains how to do it properly. Now what we've been doing is just streaking a plate, fairly simple. But if you look on page 63, they go through how to do it to where you can actually isolate bacterium from a specimen that has a lot of bacteria in it. So what you would actually do, really do it, if you look on page 63, the picture, you actually streak a quadrant, okay, and then you will actually clean, okay, with what we would use, since we're using our plastic inoculating needles. What we would do is discard of it and get another one. Does that make sense? Okay. Now, a lot of them you've probably seen down in your kits down here in the cabinets that we have some metal inoculating loops and inoculating needles. Well, those are used in a lot of scientific experimentation because we want to be able to reuse our equipment. So what is a good way to sterilize an object that you are using fire? Okay, incineration of microbes. All right, so you'd use the Bunsen burner, run it through the flame until it was red, just like we did with our slides. You don't want to sit over the flame because metal is going to melt too. So you want to run it back and forth through the flame and let it get red. Well, once it's red, it's going to have killed all the microbes on it. Do I want to immediately put that hot loop on my microbes? No, because I'm going to kill them. And my whole point is I want to be able to grow them, right? So you want to cool it and then actually grab the edge. You look here, you notice they're grabbing the edge of that quadrant and then they're streaking it again in another quadrant. And they do that 3 or 4 times. Well, if you keep going around and around, it's eventually going to spread them out. Does that make sense, what they're doing there? Okay, so that is actually the correct form of aseptic technique. Now what we did is we just transferred. We found a colony and we transferred. So hopefully when we look at our actual plates today, we have one color, one style of a colony, right? That's what we're going to look for. That means our transfer was successful. If it wasn't successful, we would probably see a number of different types of colonies. Make sense? Yeah. Okay. What are things that might have happened if we have different looking colonies on our dish?
Grabbed 2 or 3 of them.
We grabbed more than one. What else?
You didn't use aseptic technique.
Yeah, it could have actually contaminated from the air. So there's a number of different things that can happen. If you notice here on page 64, there's 1, 2, 3, 4, 5, 6 different things that they talk about to be able to help you with aseptic technique. You need to know those things, all right? Because those will be asked probably in your practical. Excuse me. All right, so that gets us to what we're doing today, which is actually answering the questions from the second part of the lab. We understood what we did is we isolated one type of bacterium and transferred it onto a plate, so ideally we should see one bacterium. Okay. Make sure there's no confusing questions on here. Okay, those questions look pretty straightforward on the back of page 71. Number 5 says, what was the purpose of the controls? Answer that. What type of control is this? You can mark through that. I don't want you to go through and do positive or negative control. Okay? Essentially, a positive control is something you would expect to change in. A negative control is something you would not expect to change. So what do you think your control would be? Do you expect a change in your control? So it is a negative. Okay. All right, let's take a look at lab 5. Everybody get that one in front of you? All right, let me kind of go through the introduction right quick because next week we're going to take the whole class break to do gram stain. All right. The introduction, it talks about cells. You'll notice on page 75 they have a cell and then they have the structures. We need to make sure we know those structures. I want to say somewhere in here that it says cilia is part of a prokaryotic cell. It is not. Make sure. Okay, cilia is only on a eukaryotic cell. Oh yeah, there it is on the picture. You see that it says cilia on the prokaryotic cell. That is wrong. Correct. Okay. Cilia are not on prokaryotic cells. They're only found on eukaryotic cells. Okay, that is fimbriae. So when you're studying those two structures, you know to make sure that it's not cilia for your prokaryotic cell. All right, let's go ahead and look at page 77 where they're talking about morphology. Okay, now morphology are the differences in the structure and appearance. All right, we have what's called colony morphology, which we've looked at in our Petri dishes, right? Different shapes, different colors, different textures, different size of your actual colonies. You notice that they looked different, okay? That's colony morphology. Now we also have cellular morphology. There's a number of different ones, and these are so hard to read on these print-offs. You may want to look at it whenever you actually look online because it's a little clearer, possibly. If you look, you should have your labs. I'll see if I can print off an original. I don't think I have the original on there. Do I have a copy online? Or have y'all paying attention to it? I'll look again so you can actually read them and see, because you want to be able to look at it and be able to identify those, okay? I also have a short video. If you have does anybody have the online version of the textbook? Because some of you have the Mastering Microbiology that came with the textbook. If you bought it new, you do. You can actually go online and look at chapter 4 when they're talking about classification of bacteria. They have the same video I'm going to show you guys. All right, 5-minute video, doesn't take very long, but once I get through with staining, I'll pull that up and we can take a look at it. Go through and know your shapes. That video is going to go through and explain the shapes. I'm not going to sit here and do that twice. I know y'all can listen. Eukaryotic cells, it goes through the organelles on page 78 and the functions of those organelles. Now, page 79, we talked about the 3 important things, or the 3 important reasons for running your slide through the through the flame, right? Okay. They're listed on page 79 as well, just in case you didn't write those down or you didn't remember them, right? So what is one of the reasons?
Kills it.
It kills it. We want to kill the bacteria. Okay? What's another reason?
Securely attaches the bacteria to the slide.
It's going to attach the bacteria to the slide, so when we're actually pouring our stain over it, we don't have to worry about it running off the slide with the stain. Okay? The third reason. Yep, it's going to cause it to be easier to stain because you're going to basically dry out all those components and then they're going to absorb stain more easily. Okay, so those are your 3 reasons. Now there is difference between these different types of stain. We're going to do simple stain today and we're going to do negative stain today. Okay, simple staining is a very, very easy technique, and they go through the differences of simple and differential versus negative on page 79, and I'm going to talk about it too as we go along. But let's go ahead and go to experiment 1. Get out your simple stain that you want to do. Okay, so you're going to need to get your crystal violet. Let's look at the actual materials. Crystal violet. You should have some deionized water in there and a dropper. I'm going to cheat because I'm not actually using a slide with any sort of bacteria, so I'm just going to use the distilled water that's right here. You should have deionized water and a dropper, so that way you can grab your water up and pull it out. Let's go ahead and get those things out, guys, since we're doing this together. Okay, so I'll get it out. And go ahead and get the one for the negative stain as well. Yeah, you should have deionized water. I'm looking on page 80. 80? Crystal violet, your deionized water.
You may put it down real quick so I can remind.
Yeah, you can go ahead and get your stuff out.
Sorry.
And I'll just walk by, y'all can go ahead and pick up your hand because we use my
What am I getting out?
This the water thing.
It looks like this.
Yeah.
It's a big bottle. It's a big bottle.
Do we need that box now?
This is? Yes, it's got the dyes in it. Where do we have extra droppers at? I guess I'll take mine over there, some video in
I feel like
here.
I'm floating there, so I do apologize. I'm like, I don't think I can possibly take any more medication. What number are you? 3113?
So this, so it's this thing right here? Structures of microscopy?
Uh-huh.
Cool.
And you want us to
There you
pick
go.
up the simple and the grant or the negative?
So that's all we're gonna need right there is these things.
We're not doing gram stain page, we're doing the other thing.
Just negative and simple?
Jessica, you need a key? Or you, she has it, oh yeah.
I claimed you today. Yeah.
There's a box in there that should have all your dyes. Some of you may want to put some gloves on. Well, by the end of this lab, I'm probably going to be covered in stain anyway, so.
Set this right here for a minute. No problem.
Is it on? Hopefully. Okay, so everybody's got their slides. You got your staining tray. You're going to set your slides on the wire basket. If you're doing it at home, you probably need to find something that you're going to put it over the top of.
It's got a perfect beard.
Just remember, if you do this in the sink, because I know you're going to do this at home, if you do it in the sink, you're going to stain your sink. Do something. I'm just making sure. I don't care if you, I don't care where you do it. If you do it outside, wherever, just make sure that wherever you do it and wherever you're putting your stain when you discard of it, it's not something you mind staining, because it stains real bad. Okay, it's called a stain for a reason. Okay. So we've already prepared our slides. We've already got that ready. So we're going to start where it says stain the slide on page 82. Simple technique. Alright? So this is over a sink or disposable plastic container. So we're over our disposable plastic container. Okay, guys? What you may do is just plan to throw whatever you're going to use away when you get done. Saturate the bacterial sample with crystal violet and wait 1 minute. Some of you have timers in your boxes. That's what those timers are for. Or you may have someone that has a stopwatch or someone that has their phone. I don't care, guys. If one person in the lab group wants to set their timer, you need to set it for 1 minute. So I'm going to give you guys a second to get all that ready before we stain our slide. Right here's where you can catch me. Okay, so 1 minute is what we need. So everybody got their timers all out. One person from the group, because we're literally doing this right now. Got your crystal violet out. Got your slide that you said is simple. Okay, you put your S on it for simple. If you're sitting here looking at me, I assume your group's ready. If not, then I assume you're not. You should have a dropper in it, because you're going to use it again. I'm sorry? You stained one already? You just prepped the slides. I don't think anybody stained them yet. So everybody's got their simple one, the one that's got the S on it, and you're ready to stain them. You already stained yours? Yeah. Awesome. Then you're good. You already stained yours in your group. You're good. Did you already do your crystal violet? Okay, good. Some people felt comfortable doing it, some people don't. And if you have not done it yet, then y'all are good. Y'all just sit here and wait. Yeah, you're good. You're just sitting there and wait. Y'all are ahead of us. You did everything, so. All right. Oh, you're good. But you want to go ahead and have your gloves on because you're about to do the negative stage. So you guys that have already done your simple stain, get out your negative one and don't do it yet because this one's kind of tricky. Okay, you can get it out and set it on your tray and be ready. All right. So those of you that are doing your simple stain, you've got them out. You're ready. If you're looking at me, I assume you're ready. Okay, so what you want to do is get your dropper, and you don't want to put, they say flood it, okay? So you're just going to saturate the bacterial sample. So you've got 2 circles, right? Yep. So in your 2 circles, you're going to go here and you're going to drop the crystal violet on those 2 circles. Now your timer needs to start once you drop it on there, okay? So your timer person needs to set it for 1 minute. Then you need to set it again for 30 seconds so you can reset it and do 30 seconds. Once the 1 minute passes, and I'm just going to say my 1 minute's passed, I'm going to rinse with deionized water for 30 seconds like it says. So you're going to get your deionized water and your dropper, and you're going to continue to do that over and over get the dropper and do that for 30 seconds. Okay? Just like this. Okay? Once you're done with that for 30 seconds, this is where you need your gloves because it's all over the place. You'll have your bibulous paper, which is down here. 1 minute. It's on the page. And I'm reading. I'm literally reading the directions. Alright, then you take this bibulous paper you've got in here and blot it. So literally, take it, put the slide in here, blot. Don't wipe, blot. Okay? Now you've got a little bit of bibulous paper in your kit, so it's saying cut the bibulous paper, fold it, blot it. All you're doing is blotting the excess water. Leave this and let it sit, and we're going to go to the negative slide. Okay? The directions are on page 82, 24, 25, and 26 at the bottom. I'm going to give you guys a few minutes to go ahead and go through that.
So we need more than one dropper, right? We need one for the water. I just use the same one.
Yes. Sorry, I'm really not live right now.
You haven't had your coffee.
Oh, I've had 2 cups of coffee. I've had Spark. I've got Maxim allergy pills and Sudafed for sinus allergy and Flonase. And I can't remember what else I've done. Oh, Mucinex. Yeah.
We got this.
I'm about as loaded as I can be. It is. I promise it is. All right.
You said you had that paper in that kit?
You have the bibulous. You have a piece of paper. It's a bibulous paper. Yeah. Should be in your structure and microscopy kit.
So after 1 minute, do we wash it off?
But you have bibulous paper down here.
We don't have any in ours.
Okay. Someone asked me a question.
Oh my god.
After 1 minute, you gotta wash it off with some water.
Yes. 24, 25, 26 is the question. The directions. I promise. Just do it just like it says. You're gonna rinse for 30 seconds with the ionized water.
Okay. Go ahead and put your paper in here. Okay.
I'm gonna give you guys a few minutes to do that.
Are these mine? Yeah. Oh, okay. But you don't miss it. Sorry, I'm late. Yeah.
What?
Drop this on there first and do it.
Drop it on. And wait a minute. There you go. Just go for it. You don't need any more, so you're done with the dropper on the Crystal Violet. You can discard of those. Now I've got your negative one. So you roll that.
I'll just have to remember what we're doing.
I thought we had to repeat again.
Sometimes it depends on the years and how fast it is.
Alright.
I thought so.
Catch me up. Yeah. Thanks, guys. I appreciate it. So what did you do first? Yeah.
And mine's simple thing. Like, well, yeah, because you have the problem.
Just the inside. But you mean your actual thing, that's like
Just gotta put it right
If it
in.
did, you don't see purple anymore.
Yeah, there's not any blue.
Done.
Next thing, like after we do the simple, are we supposed to do another one?
Wait, and then I'll go through the negative. I don't know how much I'm really helping with this matter.
Interesting.
All right.
Just put it in between.
So just, yep, just open it up, blot, put them on there, blot. Don't do it too much. Yeah, be careful. And then just set it down. Yeah, just sit down there. You're good. Ma'am?
I just messed up. I really do that.
Uh-oh.
So I put them upside down. They went upside down.
Oh.
So flip it over and now and then I wiped the bottom of it. He wiped it. And he wiped it.
Well, whenever you put your on the side, push your negatives out. Alright, now leave those on your tray, but kind of set them over to the side. Your simple stain. I got your simple stain done. Now we're going to do the negative. So get your one that says N on it out. Okay, you have one that says N. And if you notice, you've also done an extra slide in your kit. Okay, you need to grab that extra slide. You can also go and get underneath your get a slide out of your cabinet. Because you're gonna need that extra slide
In your box. I'm talking about your side.
Okay, so you go to your added slide. You need nigrosin. So you should have in your kit a tiny, tiny white bottle, little bitty white bottle that says nigrosin in your kit of stains. It's a little bitty white bottle. Okay, can I borrow that for a second from you? So it looks like this right here. Okay, little bitty white bottle. You're only using this once, so once you get done with putting your stain on this time, you don't have to worry about the nigrosin anymore if you're worried that you're going to run out. Okay, you're only using it for one slide. Now, let's take a look at the directions and read through all these directions first. Again, a sink or disposable plastic container, it stains, this stuff is black, and it stains, it is ink. Okay, you guys ever used calligraphy? Ink? Yeah. Alright, so the directions start on page 84. And it says place the clean slide at about 45-degree angle once you have actually put the nigrosin on the edge of the slide. So, here's one of the differences that you're going to do. Now, before, you drop the crystal violet directly on the bacteria. You do not put the nigrosin directly on the bacteria. Put it over on the side. Alright? So if you use a frosted slide, this might be handy because you're going to go, okay, over here on the side, I'm going to drop 2 or 3 drops of nigrosin on it. Okay? Then once you finish doing that, you're going to take your second clean slide, put it at a 45-degree angle, and literally draw the nigrosin across. So here's my nigrosin right here. Okay, I'm going to put it here. And then I'm going to literally take this and slide it across. So I'll go ahead and do that for you guys so you can see what I'm talking about. Alright, so I'm going to put like 2 to 3 drops on the side. You don't have to put it directly on it. I just did that because... this is my negative slide. So say my bacteria is on this. Okay, I don't want to put directly on the bacteria. So I'm going to take it at a 45-degree angle. Hopefully y'all can see this. I'm trying to make it so the video can see it too. And then I'm going to draw it across the slide. Now hopefully you put them all the way down. I had mine sitting up at an angle. You want to put it all the way down and draw it across the slide. Okay?
On the bottom side?
Yeah. And you just draw it across the slide. Okay? Now it says, make sure you got your gloves on. Okay. Says air dry the dye stain sample for 10 minutes. So you are not rinsing this off. Okay? This one, you let it sit. It should dry. Okay? You're not turning these in today, But when you're done, I expect for you to go through, observe them under the microscope, fill out the chart. According to what I have over here, it says fill out the chart on page 83. That's for the lab experiment 1. And then answer the questions number 1 and 2. Okay? And then for lab experiment 2, fill in the chart on page 85. Answer questions 1 and 2. Let's go ahead and glance at these questions right quick. On page 84, it says, what differences would you expect to observe when changing the magnification from 100 times total magnification to 200 times total magnification? What would you expect? Okay, more clarification because you would be seeing it at a greater, so you'd be seeing maybe fewer organisms, but you'd be seeing it at a more detailed clarification, right? Does that make sense? What about 200 to 600? Same thing, right? Okay, even more clarification. All right. Okay, so what do you think we're doing with the crystal violet? Number 2 says, how does crystal violet enhance the visualization of bacterial structures? What do you think we're doing here? You're enhancing the actual structures so you can see them better. Now, the negative stain, here's the interesting thing. Okay, most stains we use are positive. So what do you think a bacterial cell would be when we deal with charge if most stains are positive or want to see the actual inside of the cell? They're negative, okay? So what do you think is going to happen if I put a negative dye on them if they're negative? They're going to repel, right? Okay, so the whole point of a negative stain is to look at the outside cell structures because we are going to be using a negative stain, which is a negative charge. Okay, what is a bacteria as far as charge? Negative. Okay, so what's going to happen is it will repel it. Okay, so instead of seeing the internal structure with the negative stain, you'll see the external structures. Make sense? Okay. So let's take a look at the questions on page 86. It says, how does nigrosin work in the staining process? It's a negative stain. So it's going to repel. What is it staining? The outer structure, right? Which is the cell wall and the slime layers and things of that nature, right? What would you be able to notice more with the negative stain? The outer structures. Make sense? Okay. That's the interesting thing about those. And if you read where it first starts to talk about the experiment, when it says experiment 2, negative staining, there's a couple little 3 or 4 sentences about it. That's where I'm gathering that information from. I promise I'm not pulling it out of the air.
What page is that?
Okay. That information is on page 84, where it says experiment 2, negative staining. Very important to read through those little paragraphs when they start them. Okay. And read through your introductions for your lab. Alright, so at this point in time, I want you guys to go ahead and go through your negative stain. Wait, just a second, are there any questions about your negative stain? It's pretty simple. It's pretty easy, I should say. Alright, so y'all go ahead and do your negative stain. You need to answer the questions on page, I've got them all written up here. Complete your chart on page 70, and then complete the chart on page 71 and answer the questions. That's for lab 4-2. Turn in lab 4 up here at the front by my microscope. You need to discard your dishes, same place we did last time, in the sink over here. I'm going to get the sink bath done. Okay? Look at the slides that you prepared for simple and negative stain, and then complete the charts and the questions on page 83, 84, 85, and 86. Okay? You get that done, you're free to go. As far as I'm concerned on your slides, you need to look at them in the microscope by next week, okay, and get those answered. So if you want to keep them, do that next week. That's also fine if you run out of time, okay? Should have time to get it all done. Should, okay? Any questions?
How long do we need to let them dry before cooking it? 10 minutes for this one.
Yeah, well, what you're gonna do, guys, whenever you get done with your staining, go ahead and do lab 4 too.
Yeah.
Get that done and out of the way. By the time you get that done, turn around and look and make sure everything's completely dry, and then you can go through and observe. Now, what magnification do you think I want you to use to observe them? Oil immersion, the high one, okay? So you're gonna go through your steps, go low, then go up, and you're gonna practice oil immersion today, all right? Anything else? All right, I'll let you guys do your negative state.
Are we done?
We are done. Stop that.